All right, so welcome back to Data Science Omics 2020. Today, we are going to talk about handling large and complex data sets because ultimately our goal is to actually start looking at the conditions that we are trying to study. And the data set that we have is not that simple, right? It has a lot of noise, it has a lot of additional types of information. So we'll take a look at a few examples. Hopefully we'll have enough time to make significant progress today. But before we do that, let's take a look at how everybody is doing. As we did last time, I was just looking at everybody that did some additional coding exercises and ran or started the pipelines. So one of the things that I wanted to show you as we continue is what is kind of happening behind the scenes. So as you remember, yesterday we started all of those pipelines. And in the context of bioinformatics, what a pipeline does is you prepare a script that puts together your data, puts together the settings for all the individual jobs, and then you can deploy those onto a particular environment. And in this case, we have an environment set up where multiple servers are performing a variety of jobs for many different users. So here you can see the list of processing pipelines. So these are the processing pipelines, and then the jobs are actually either running, or if a particular server does not have sufficient RAM or CPU that was assigned to a job, it is going to be in queue. So probably some of you are recognizing your names here, because as you can see, the majority of you that started the pipeline, it has not finished yet. And so I just wanted to explain how that happens. Now, one of the important parts of this is that pre-processing jobs actually take up a lot of RAM because what they have to do is they have to pre-process all of the reads, individual reads, and store them in a new file. And so these files are huge. And if you have multiple samples, that file for each sample takes up the space and takes up the server. So practically speaking, none of the downstream analysis can actually process until these are complete. And so we'll wait a little bit longer, that's fine. But I just wanted everybody to know, you should probably see anywhere between 50 and 66% on your pipeline. Okay, so what did we talk about last time? Last time, we started talking about statistical properties of data. So we looked at distribution, we looked at correlation, and how to apply these to understand what our data is looking like and how do we prepare it for downstream analysis, which is what we'll do today. We looked at summarizing this information visually, where we used log normal transformation to make this more of a normal distribution and to have more information visible, and also to enhance the power of our parametric test. We also talked about distribution and what parts of the data are actually represented on a distribution. So distribution for a particular sample could be transformed by both logarithmic scale and filtering. And one of the important things that it gives us is greater power in distinguishing noise from signal. So as we are looking at conditions, as an example here, we looked for genes that have high confidence. We have high confidence that these genes are actually different in terms of their significance overall, and not in any particular individual sample, right? Now, after normalization, we have both greater confidence in the differences that we observe, and we have greater confidence that the samples that we use in conditions actually represent replicates of that condition and don't give us too much noise. And we also looked at how to do this in R, looking at different examples of bar plots And I hope that all of you also had some time to practice it independently, both in R and in Python. And the assignment that was posted, right, we reviewed the assignment from the previous time where you were supposed to, so by the way, these assignments, I just want to show you where they are. On the forum, right here, I pasted these assignments, and hopefully all of you are able to see them. So I only saw Marek answer. So I want to ask you real quick, did anyone else have their assignment? Was anyone else able to do this assignment to actually find gene 1 and gene 10 and kind of see the bar plots and consider which one is more informative after what we did yesterday? Did anyone else try to do this assignment? Oh, okay. Well, you know, if you just even post this kind of a response, just saying, okay. So Adolfo, just like this, for example, you can mention that a particular gene is more informative because the differences in these samples or those samples are actually, you know, more or less interesting or something like this. So if you could post it here, then I'll be able to respond to anything. Okay, and then Janani, you found the genes but couldn't find which one was informative. So last session we talked about this. Were you at the last session? Let's talk a little bit. Okay, great. So again, if you could, did anyone else have trouble doing this assignment or finding the genes or kind of plotting them? Yeah. What were the issues that Samitha and Hadiya, what issues did you come across? Okay. So maybe try to do this assignment. Basically, the code is available. Bibsa mails every day, I believe, the link to an updated, okay, downloading R and Python. So Smitha, have you tried it on the code.omicslogic.com? Okay, did you get the results? Okay, so here you can see a blog post on understanding the basics of R, and also if you go to here, well, actually to this link right here, to about, you will find information on also how to install Python. Okay, so please take a look at those, and there are also some packages that we will be using. And basically the way it works, if you follow the specific, so we are following these specific exercises. Okay, so here, for example, we already did this one, we did this one, and we are gonna do this one today. So these are the three that we're doing, and then next week we're actually gonna do these three. Okay, if anyone else has any issues, again, I encourage all of you to post these right here, specifically if they're about coding exercises as well. So those were the assignments that we reviewed, and to kind of illustrate what the results were. If we look at a gene like this, when we look at it visually, the information is in here, and the information that we have is not represented clearly, right? It is there. We kind of see some kind of a pattern, but it's not very clear. And so what we can do is we can sort the data. And now when we sort the data, well, this is for gene 6, but we also did it for other genes. We can start seeing, for example, this gene 6 is informative because why? What is the information that this gene actually contains? Exactly. So Adolfo, that's correct. Well, it's not differential expression, right? We're just exploring one gene, but you're correct. It has low expression for all the cloud and low, and then high expression for all the rest. So if we wanted to kind of see what is the information that this gene contains, that's the information that it contains. Now, we also did correlation by checking whether the samples from the same type, for example, normal-like versus normal-like, or luminal versus normal-like, right? Not the same type, whether they correlate. So do you think that these two normal-like samples are good replicates to represent this normal-like condition? And if yes, why? And if no, then also why? Right, exactly. So when we look at this type of a visual, what we want to do is kind of draw a line. And if it follows that line, that means that if it's one here and one here, that's the same or close to the same, right? If it's two here and two here, it's close to, right? So this means that these values are closely correlated. And so what we can also do is multiple pair testing, and we did that in R. And here we can see for multiple comparisons, for example, normal-like versus normal-like, or normal-like versus basal, or normal-like versus another basal. So hopefully, if you follow along, you will be able to see the portion of the script that we use to produce this graph, and also gives you the actual correlation values. And after that, we had two more additional assignments. Right, normal to normal correlation means that the samples are not too different from each other, right? And therefore they're reliable representatives of replicates, because we'll talk about this today when we talk about uh, studying conditions that these samples represent. We know that if we want to, let's say, compare short people to tall people, if we take a bunch of short people in the US and a bunch of short people, let's say in the Philippines, we might find that the different definition of short might make a lot of variability within each group. And so if we do that kind of a comparison, we will see that the condition that we want to represent, which is shortness or the height of the person, is actually is noisier when we have variation inside the group. And so this kind of correlation in terms of all the genes that we have helps us see that it is a reliable selection for us. So the assignment, I don't know, did anyone attempt this one to load the full gene expression table and compare specific genes? Did anyone have a chance to think about this problem? Okay, so maybe for this assignment one, what data set do we use? Okay, so the data set that we use, again, is if you are doing this on your local R, in your local R studio, if you look inside this data folder, right, this is the link that I think Beeps has shared with everyone. it has all of the scripts that we're using and all the data sets. So the full data set, right, we used up until now the 15 gene. So we looked at the 15 gene, and this was kind of a simplified table for the 15 genes because the genes are not real genes. They're just gene 1, 2, 3, and 4. And then the names of the samples are not real named samples. They are all specific type. And then we looked at gene expression data where we actually have ENSG, which is ensemble ID. We have the name of the sample, and then we have the type of the sample like this. So this would be the full data set, gene expression data. If you want to have a smaller data set, this data set, we explain how this contains information that comes out from the pipeline. So this actually includes the adapter elimination and PCR cleaning and the pair end for each one of your sequence read archives. So this is an ID of this sample. And then here you have the raw kind of gene output for all of your pipeline analysis. So you can use either one. This is in FPKM, and this is in already log transformed, normalized. Okay, so any questions about these assignments? If you don't have any questions, then if you could complete these for our next session and post your answers on the forum. Okay, so any questions about these? Does anyone have any questions about the assignments or where to find the files? Okay, so if there are no more questions, this is what we want to cover today. We want to talk about hypothesis testing, differential expression analysis, factor regression analysis, and then see how those are implemented in a package called DESeq2. And this package, by the way, also works under Bioconductor. So the reason we're talking about these is because overall in the real world, right, when you are dealing with a bioinformatics omics data set, whether it's gene expression, whether this is a metagenomic sequencing or variants in your DNA sequencing, you're going to first of all work with some kind of data that has been collected in an experiment. And based on that experiment, remember we did data pre-processing and cleaning. And so what we typically end up with is data that contains, for example, additional sequences in the reads. And that could be for genomic sequencing or for RNA sequencing. We can have different types of sequences that we first have to prepare as operational taxonomic units if we're talking about metagenomic sequencing. So whatever that process of pre-processing is related to our experimental design and how the data was collected. And we also need to clean up that data. So a lot of times this data could include missing values, low expressed genes or OTUs, or variants that have low significance in terms of our overall sample. So we do a lot of data cleaning, and that was really the objective of the first part of the program, is to just get familiar with some of these methods for cleaning the data and preparing that data for analysis. And then we do what we call exploratory analysis. Exploratory analysis gives us a sense of the things that we discussed yesterday. Are these samples comparable? Is our data normally distributed? What are some statistical properties of the different samples? Do we have any outliers? Do we have any kind of strange values that do not make sense? How do we explore all of this? We do exploratory analysis using visualization and looking at statistical properties. And oftentimes that leads us to a variety of different questions about the data that we can explore, and that's called data mining, when we start asking questions of the data. So we need to start translating our questions. Oh, why is this sample different? What is the information that this gene contains? How is this set of genes characterize difference between different types of conditions? So that process is what we are doing now. And then your objective is to actually start selecting a model that represents your data. So we'll talk a little bit more about that today. and we'll then continue talking about machine learning and more advanced methods in our next week. Now, one of the things that we kind of went through, I think, is in all of these sessions, Janani here is asking, since new files are added to the Dropbox frequently, is there a way to save the data folder to my computer such that it periodically updates when you add new files? So yeah, that is, you know, then I would have to share the full Dropbox folder with you. For those that actually want to do that, you can follow the Git repository if you would like to. It's going to change. So after this program is over, I'm going to delete everything from there. But if you would like to, you can actually subscribe to this Git repository, and then you'll be able to follow it. I think this is, yeah, you can watch this repository. And so you'll see all of this updated here. Okay, Janani, do you know how to do that? Watch this folder. So you'll see updates there. Okay, so we talked about how to make sure that the data is clearly presented in the course of the first few sessions. For example, we talked about sorting our data and making this data organized by type and summarizing it and then visualizing it as a table. We also looked at plotting different conditions and using that visualization to compare. We also talked about scatter plots, and we'll continue talking about scatter plots. But the objective of these exercises is to really start thinking about, what does this data represent? So ultimately, the problem that we are trying to solve is that we have some kind of a condition. Let's say in the case of this last few days that we looked at cell line data, what are the conditions that we are studying? Who can tell me? Okay, so yes, different so these are actually cancer subtypes. They do include a normal-like, but that is still a subtype of breast cancer. It's just that the expression of those genes is very much like normal, instead of normal tissue, instead of some abnormal tissue. But these are all subtypes of breast cancer. And so when we look at this kind of data, we're looking at only a small number of samples that is supposed to describe some population of cancer patients, right? So we are using this data set because we are trying to do what's called inference, statistical inference. We found some pattern, and we have some confidence that this pattern is going to help us understand this condition in general. And the ability to eliminate noise in the data that we have is very important, because if we want this to be representative of the population at large, we don't want to confuse the noise in our particular data set with something that will not be apparent. And so that's why this problem of confidence and statistical confidence in the observations that we have. And so one thing that we can do to understand the differences between these two conditions is to ask a question. Well, if I have a hypothesis, I have two subtypes or two types or two conditions, can I actually determine whether these conditions are different? And if they are different, what are the specific differences that I have high confidence in? So to do that, in our case, we're going to turn to a number of two actually examples. And one example is going to be subtypes of breast cancer. Another one is going to include some additional variation related to how the data was generated, because it's not going to be generated from cell lines. It's going to be generated from animal mouse models that have tumors engrafted in them. So as you remember, the data that we have is a table of expression. In this table of expression, we have a number of genes or observations about samples or objects. So these are the objects in our population, and the genes are the observations, and the data that we have about this, each one of our observations is the expression level, right? So the level of expression is supposed to represent the difference in this condition. When we look at our profiles of genes, we can see that some of these profiles can be very noisy. Okay, so this is a little bit of a different data set, but it's a very similar idea. You can see here two genes. This is a human gene, ANSG. This is a mouse gene, ANS-mus-G, so from mus musculus. And you can see here in your description of the samples, you have ER positive and triple negative breast cancer. So two conditions, ER positive, triple negative. Looking at this data, looking at this gene, one of the observations that we have, what do you think is happening with this gene in these two different conditions? What information does this gene give us about these two conditions? Okay, so Samantha, to your point, triple negative, right? You're saying triple negative has high expression compared to ER in triple negative samples. But if you were to take these samples, right, they also don't have any expression. Or if you want to take this sample and that sample, they have very low expression. So how can you be sure that this is a pattern, not as a result of some random variation? Okay, that's the question that we're trying to ask. Or if you look at this example, in this example, maybe it is even more difficult to say what is the actual difference. So when we talk about differential gene expression, we're trying to say that we want to identify only such genes that actually represent a pattern that we have a high confidence in, that represents some difference between these two conditions. And so some of the challenges for differential gene expression, okay, we could cluster them. We'll talk about that in our next, in one of our next sessions. But for now, we just want to use statistical analysis to determine what is noise and what is the specific signal that we would like to look at from a biological perspective. So here we have thousands of genes expressed in a typical sample, maybe 20,000 like we saw in our previous example. In this case, we have two different genomes, right? The mouse genome, the human genome, so it could be as many as 60,000 genes. Samples in each group can have significant variation in levels of expression, and determining a threshold for significance is a major challenge for these differentially expressed genes, because as we saw, many factors influence that. The correlation or the variance inside each group, as well as what do we call biologically significant. If we have 5 and 1 or 2050, what is biologically actually relevant? So to talk about this, you're probably familiar with William Gossett. William Gossett was a statistician that worked for the Guinness Company making beer, and his statistical interests were in sampling looking at statistical significance of small sample sizes. And so one of the things that he did is he published this paper called The Probable Error of a Mean. And as a result of that paper, today we have what is called the student's t-test. And the t-test basically says that if you take your samples in a random way, if you collect your samples in a random way, there's going to be this t-distribution that represents what happens by chance, right? So if it's a random sampling, this represents the values, distribution of your values that are completely random. And as a result of looking at the difference between signal and noise, or the difference between group means and the variability within each group, we can actually use this formula, this test, to select the non-random observations. So for example, when we do this kind of an analysis, the outcome is a p-value. And the p-value is really the area under this curve. So the area under the t-distribution curve, in the case of the t-test, is going to give us the unlikely observations. And we can have some kind of a cutoff. For example, we can allow ourselves to make small mistakes, right? So maybe in 5% of cases, we might allow ourselves to make a mistake. And so that is where this number p-value of 0.05 or 5% really comes from our threshold, or allowing ourselves to kind of make these small mistakes. Now, that's not the only problem. If we only had hundreds of observations, that would be sufficient. All we need to do is to determine a p-value, and so t-test is a perfectly good way to extract statistics from our comparisons. How do we extract the statistic? What is the statistic that we extract from a t-test? Let me pose that question to you. What would be the result of this kind of a test? Okay, that's one part of it. And what's the second part? Again, let's look at this formula right here. What are we actually, so the p-value, the confidence level, and what? So confidence in what? What are we confident about? Just look at this formula right here. Confident in what? That there's a difference between what? Group means. Excellent. Yeah, so difference between group means. So we are looking for the difference between group means by looking at all of the differences and selecting only those that we are confident in that they happened not by chance, not a result of some random noise that could have come up by random, by chance. Okay, so the problem with this many observations that we have is that if you have a very large number of observations, by chance, you can find statistically significant ones just because of this family-wise error rate, and actually select the ones that are going to be false positives. You could also have false negatives. And so when we have this kind of a problem, the typical approach is just to increase our threshold for confidence. So for example, instead of having 0.05 for your p-value, you can have an adjusted p-value. And that adjusted p-value could be the Bonferroni, or you can have a number of different types of more stringent approaches to the p-values. The problem with those is that they actually do not specifically address the challenges in these kinds of RNA-seq, for example, experiments. And so not so long ago, the false discovery rate approach was actually published by Benjamini et al. And they are saying that basically the way we can approach this to minimize false positives and minimize false negatives is to do a fairly simple procedure. And the actual procedure is not the discovery here. The discovery here is that this procedure actually seems to work all the time. And the approach is that you can actually put all of your p-values in ascending order. So it means that from lowest to greatest, and then you can assign ranks, and then you can calculate each individual's p-value critical value, which takes into account the total number of observations. So that means that this approach is linked to how many observations you have. So what are the observations in our data set? What are the observations that we have? No, cancer types are objects, or they're conditions, sorry. Genes, right? So observations are exactly their expression levels of genes. That's our data. And if we consider the number of genes, right? So for example, this is false discovery rate, or FDR. FDR for gene set of 60,000 versus 20,000 versus 15 would be very different, right? So at 15, it would practically make no change to the p-value. As your number grows, that number will make your p-value calculation of false discovery rate larger. So that's the major innovation here. And so when we look at this typical analysis of, for example, RNA-seq, we want to make sure that the input data is good, and then we obviously map it to the reference genome, we quantify expression levels, and at the end, we actually have to do differential gene expression. So calculating differential gene expression would apply a lot of these methods to make the statistics of finding a mean, significant mean between groups of samples reliable. So let's take a look at how to do that first on the platform. And I hope that we can all have, hopefully all of you have your data already saved, and the newer data that I asked you to download, or Beeps has sent you an email to download from that folder. We can either add this at the end of a regular pipeline, or actually we can run this completely separately. So let's do that separate option, where we will find a number of different ways that we can look at the results from a differential expression pipeline. So let's go to the platform and let me know when you get here. And by the way, the gene, if you want the direct link, or the data set that we will use is this one. You can just download that if you don't have it. Let me know once you do have it. I'll actually give you a couple of these so we can try multiple things. Okay, so please let me know once you get to here and you go to differential expression. So this is the link. Okay, just say yes when you get here. Okay, anyone else? All right, great. So let's upload our data set right here. You go to File, Upload, and we're just going to take the data that came from that PDX study. Okay, so here it is going to be called PDX HT-seq gene expression. Okay, so this is important to remember, that we're actually using data that was processed using HT-seq. Remember, in our last exercise, we used our SEM, and we selected for FPKM values. And unfortunately, FPKM values are not going to be the appropriate data. You can see it right here. Please note it is recommended to upload the expression in raw read counts. And the raw read counts, the fastest way to generate them is using HT-seq. Okay, so now group selection. When you press on group selection, all of your column names, remember this is your header, appear right here. And the first two letters of each sample represent the condition that we have. So we have 1, 2, 3, 4, 5, 6, 7 triple negative, and 1, 2, 3, 4, 5, 6, 7 ER positive. 1, 2, 3, 4, 5, 6, 7. Yeah. Okay, so you just drag your one type in one group, because this is going to represent one condition, and your ER positive in another group. And click on submit. And let's select, and you can select either one. I'll select DSeq. You'll see here a few different parameters. Okay, so let me know when you are here. Okay, let's take a look at some of these parameters right here. Significance threshold, p-value for differentially expressed genes. Threshold for log2 fold change. So log2 fold change is the fold change is the actual difference between means of these two groups. And it is going to be in a logarithmic scale. And you can see that this is going to be used for visualizing the volcano plot. Database human, right? So we're just going to take the human genes here. We can do the mouse gene separately. And significance threshold for visualizing enriched KEGG pathways and GO terms, right? So if we can, let's increase this and we can actually keep that like that. Well, let's do 0.05. Okay, and click on save. And run your pipeline. Okay, so maybe type in pipeline when your pipeline is done. Or if you have any questions, let me know. Okay, which part do you need me to repeat? How do you know how to get inside this section, Calamari? Okay, so you go to areas of analysis and right here, differential expression. Okay, so let me know when you're here. Okay, so here you upload the file. Do you have the file? All right, great. So you go and you upload it right here. It should say PDX HTSeq gene expression. Choose, and then it's a little bit of a big file, so we'll upload. Okay, let me know once it uploads for you. Yes, we changed the default parameters. I mean, you can keep them the same. I just changed the p-value to 0.05 and the other parameter at the bottom there to 0.9. All right, great. So now group selection. And you should see these right here. So you just drag all of the ones that say TN, triple negative, into one group. And all of the ER inside this group. And then you click on submit. Yeah, not only you, but all of us, right? So all of us have a couple of dedicated servers where all of this group can submit their jobs to. And once the pipeline, or once all of the jobs are processed, each one will get their results. Different jobs are in queue, not all of the pipelines. Yep. All right, so Calamari, right? So you click on start, and then you put on DESeq2. Okay, Calamari, are you here? Or for everyone else also? All right, great. So here, what I did, again, you don't have to do it, but just it's important to understand what each one here, and you can hover over the little eye, so you can see the p-value filter for significant gene analysis. It is also used for the threshold in the volcano plot. So here's significance threshold. And as we said before, we can choose 0.05, for example. So I'm going to do this. And then here, again, you have log2 fold change. So you can have a number here, and we're going to use the human. And then I just also changed this one to be 0.9. That will give us more pathways. Okay, and then you just click end, save it. I'm not going to run it. So let me know once you got this done. Okay, did anyone else? So Maria got it. Who else got their pipeline started? All right, Adolfo. Okay, excellent. So what is this pipeline actually going to do? And I'm just going to delete this one. Okay, what is this pipeline going to do? This pipeline is going to apply this differential expression analysis plus a number of visualizations, and it's the exact same job that appears at the end of the typical RNA-seq pipeline. So at the end of your preparation, you can actually connect those jobs at the end, but you have a number of different options: DSeq, DSeq2, CuffDiff, and EdgeR. And all of them, really what they seek to do is to apply this kind of information or this kind of analysis, statistical analysis, to determine which genes out of all of the genes that we have in our data set actually have a significance. They have some significant difference. The results are, first of all, as a table. So here you can see an example from results from DSeq2 and from EdgeR. So what are these lines, rows right here? These are the genes, right? And then here you have two important places to look for in your output table. The first one is log2 fold change. So what do you think minus 0.3 represents? What do you think this number means? Okay, exactly. So Maria is saying the first is smaller than second, right? So we can't say anything about it being down-regulated or, what is the characteristic. We just know that we had group 1 and group 2. We didn't specify which one's normal, which one is not normal. And so it's important to remember here that this parameter is the difference between means. When can a difference be negative? Is when the first one is smaller than the second one, right? And what is the p-adjusted value? Remember, this is actually explained right here. This is the p-value adjusted for false discovery rate. So the log fold change, and here you can see the log2 fold change. This is the log fold change. Basically, this is in logarithmic scale, the difference between these genes. And you also have information here that allows you to go back and figure out what was the actual mean overall in your whole data set, right? So that gives you some idea of whether that gene was in general high expressed gene or low expressed gene, right? If the mean is high, you can, for example, use this column to find only genes that had some significant expression. Now, the additional outputs here can be used to visualize your results. And the visualization of results includes some plots that basically all help us just focus on the right proportion of genes. And typically this is some small number of genes that we're going to be looking at. Now, what is the difference between EdgeR and DSeq2? DSeq2 is, both of them actually are going to be looking at specific normalizations first within each group before they actually do the analysis. And the other important difference is that EdgeR is typically going to be looking for a minimum of 3 replicates per condition, because if you have some condition that, let's say, has a 2 samples that are differentially expressed, and the third one is all of a sudden very different, it's going to see that as an unreliable result. So it has some additional stringency for the for consideration of this type of a difference. Now, when we look at the output, and yeah, I actually don't have, so when we look at the pipeline from edge R, we're going to have, the exact same settings here that we need to consider. And the only thing that we're actually going to have is we have the output. So one of the challenges when you have this output is that you lose your information from the original table of expression, right? So let's take a look. I actually have the same example in our folder. So let me give you the specific link. Like that. Okay, so if you look in here, it is called DEC2 PDXR. So let's open this. We can open it in Excel for now. Okay, so let me know once you have this data set. And we'll also try to find what is the actual gene expression in our original data set based on this. All right. Okay, so let me know once you have this open. Okay, and to find the most differentially expressed genes, we have to look for the p-adjusted value, right? So we can just use a sorting option. So we can sort from the lowest to the biggest. Okay, and then the other parameter, like we said, is going to be this log two-fold change, right? So here you have really small values. Let's find the 0.05. Okay, and once you find it, tell me how many differentially expressed genes do you actually have in this data set? Okay, so type in your answer. How many differentially expressed genes do we have? Did anyone find the number? Okay, so Deepika is saying 4219. Okay. Anyone else? Janani is saying 2818. Anyone else? Four two one eight. Okay, anyone else? Okay, Daisy has 5,394. All right, so how do we actually determine? Let's say we take our threshold at 0.05. You can see that the last number before 0.05 is 0.04, right? And so the row number is 4219. But how many do we actually have for 4219? It's 4218, right? So 4218 is how many, because we have the header row. And so that is the correct answer. But what is the problem here? The problem here is that I don't know what is the actual profile. What is it that I do know about this, right? So let's just highlight it. Okay, this is the last one. So what I do know about it is that this is the log to fold change, so which means that group 1 was upregulated compared to group 2, right? And it is statistically significant, which statistics only means that the variability inside the groups is sufficient for me to say that this fold change is actually statistically significant, right? You understand how those two values are used. The statistical significance is only helping us distinguish noise, which is variability within the group, from signal, which is the difference between the means of the two groups. The fold change is the actual information that I call statistically significant, right? So whenever we are looking at this kind of an output, we're really looking at the results of statistical analysis in the context of differences between means. Okay, so now let's go ahead and run this same analysis in R. Okay, so let us close Excel. Well, okay, the first thing, let's now find one of these genes and just kind of confirm that what we're seeing is correct. So let's open the original gene, or the original gene expression table. Okay, please let me know once you open the file PDX HT-seq gene expression, right? And let's take this first gene. I'll just search for it. All right, what does this tell me? What is this gene expression? What information does it tell me? The information that it tells me is that this particular gene does have at least a few examples from the total population of triple negative breast cancer patients that have it a pretty high expression, right? It's right between 350 and 400. And the ER positive, let's assume I selected by random, by chance, have none, right? They don't have any expression. So I can do statistical inference. I can infer from this analysis that this is a gene that on average, kind of like if I randomly select a person from the population, if they have triple negative or ER positive breast cancer, they will have a difference in this particular expression. So let's go to R, okay? Let's, does everyone have their R Studio open? All right. Okay, does anyone else? I'm just going to give you a couple of minutes. All right. Okay, so the first thing that we want to do is load this package called DEC DESeq2, or it's a library called DESeq2. So what we do is library. Yes, you do need bioconductor. So for those of you that don't have bioconductor, you will have to install it. The way you do it is you go to your tools, install packages, and here you will say BioC Manager, like that. Okay, so those of you that are following along, you go and you add BioC Manager. After you install it, to install DESeq2, you go to BioC Manager. Yeah, BioC Manager right here, and do DESeq2, and then it will install it. Okay, bioconductor has a lot of good methods to deal with this kind of data. Okay, so we first of all need this library DESeq2, right? The second thing that we need to do is we actually need to load this data set. So let's load our PDX data set, and we are going to read table. And obviously you need to have your working directory set to this folder. So make sure that you do that before running this. And what is this file called? This file is called PDX-HT-seq gene expression.txt. And then we want to remove, or we want to specify that there's a header, right? And we want to already create a row names, which is going to be the first row, right? So these are going to be the first column, sorry. And the separator is T. Yeah, well, the code is actually in the folder. So yeah, okay, I can do it line by line, let's say, right? Okay, so this is the first thing that we want to do, and let's just do, let's just preview our data set to see what's inside. PDX data set. Oh, that was supposed to be true. Okay, did everyone, did I stop sharing? Did everyone get to this point? Or let's see how many people got to this point. So if you got to this point, okay, great. So you can see, right, a lot of zeros there. Okay, so then we want to create a data set. Let's just call it data. We'll use that later on. So we'll use data, and we'll put our PDX data set in there. That. And let's store separately the column names. So we'll have column names PDX dataset. Put them in names. Sorry. And we also want to have our metadata, which is going to be, well, before metadata, we need to set the classes, right? So we need to specify what is in our data. So we need to say that we will just call them, right? We have, let's take a look at our data set here. So we have 1, 2, 3, 4, 5, 6, 7, 7 ERs, and 1, 2, 3, 4, 5, 6, 7 TNs. So let's just write that out. So we're going to have 1, and then we can just copy this 7 times. 2, 3, 4, 5, 6, 7, and then we'll do TN. One, 2, 3, 4, 5, 6, 7. Okay. 1, 2, 3, 4, 5, 6, 7. Yep. All right. So now we want to add a metadata and add in there the data frame, right? So we're just going to have our information inside here, and that's going to include the names, and they're going to include the meta classes. Right, so the names and the meta classes. And now what we want to do is run, or we want to define just the data set for DESeq, right? So it's called DESeq, so DS. And we'll run DESeq, DESeq data set from matrix. So this one. And the count data. So what is the actual data that we have? That's data, right? So right here. Can we use instead CER? What do you mean, like right here? You can extract that information from the actual headers, but you'll have to do a few more. Yeah, you can use, that information is in there, right? It's right here. So you can actually separate that and then get that information out. Okay, so now, just for simplicity, we want to set up this data set, which will include, right? So let's go back here. Okay, so here you can see now we need the count data, then we need the call data. So what's the call data? The call data is the metadata, right? So in the metadata is our column data. Okay, and what are the conditions? So what is the next thing that we need? The next thing that we need is the design. What is the design? Design are the classes, right? So the classes is what represents our condition. So the way we do it is design, and you have to do tilde meta classes right there. Just make it a little more neat here. Okay, did everyone get here? So I'm just gonna copy this here. All right, and then we will okay, maybe actually before we run DESeq, let's filter out because we have a lot of zeros here, right? So let's just filter that out. So we'll do DS, and we want to filter it by row sums. So we're going to do row sums. And we're going to say that inside of our counts of DS, we have more than, let's say, 2. Okay, let's view our DS, right? So we'll do view. Well, actually, let's do head. Argument 15. So we have not enough. 1, 2, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Oh, we need another. Oh, this. Count DS. More than 2. Let's try this. Oh, okay. So we actually, yeah. So let's see. Count DS. S4 object. Okay. So we can't do that. Yeah, where was I? Was here. Okay, let's do this. We will row sums. Let me just see one sec. So what was it saying? X must be a vector. So it is. Okay, let's do this. Let's perform dseek and see if that works. Dseek to data, and we'll add here dseek, so seek of ds. Okay, let's try this, and we'll get the results to make sure that we have the results printed out. So I'll actually take this, and we will add here the results from dseek to data, and we'll print dseek results. Dseek to results. All right. Okay, so here we have our data set, right? So here we have the result. Yeah, I'm not sure what happened here. I'll have to look into why I can't filter it. But if all of you, did anyone get to here and get this output for, did anyone get the output for the base mean, log to fold change, log fold, standard error, the p-value, right, the p-value? Did anyone get this? All right, great. Okay, so the other thing that we can do, all right, great. So the other thing we can do is we can actually plot this as a volcano plot. So there's another library that we can use called volcano plot. And this library is called something like advanced volcano plot, enhanced volcano plot, volcano. Okay, so you got to add enhanced volcano here. Okay, what about the rest? I see Adolfo and Krishna got it. Did anyone else? Janani and others? Did anyone get this output? All right, great. Anyone else? Ah, thanks, Cindy. Let's try that. Great. Well, actually, DSeq itself normalizes the data, so we don't have to do it. All right, so here instead of count, I forgot an S, so that's why it wasn't working. So now we got it working here. Okay, so now we have this data frame DSeq2 results, right? And the DSeq2 results, we see here the results are what? They are TN versus ER, right? And we have 878 rows and 6 columns. What did we have before? Yep, 878 rows and 6 columns. All right, so now let's quickly plot this as a volcano plot. Did anyone else? Yeah, I know there were at least a few other people here, but okay. Hopefully you can continue after. So let's do this enhanced volcano thing. So for the enhanced volcano, where right here that we were using, so when you do DSeq, you specify what was your data, what was your call data, and then what is your design. So the design, right, you remember we added meta classes here and we specified for each one of our samples, what are the classes? And so I got the ER TN versus ER right here. Okay, Hidia? All right, enhanced volcano. And we have our results, and we're going to take the log two-fold change right here. So log two-fold change. And then the Y value is going to be the P value, right? So we have these two parameters, the P value. And we want to take the row names from results, and we'll just take the limits of all right, let's try this. Okay. Unused argument P value. So what was the like that. There we go. Okay, let's take a look at this volcano plot. What does it tell us? It tells us, right? So we set -5, right? And 8 over 8. And these are the differentially expressed values. So what is this? This is the log fold change. So we can play around with this. So let's do -2 and 5. Okay, so this is something that we can play with. And let's say -1. Okay, minus, what was the original? 5, 10. Okay, you can see that this changes in terms of the cutoff for our information. So this is a useful and a standard way to visualize our data, to have a sense of how many genes in total we have and what are the most informative genes from these. Now, the final thing that we want to do is we want to take these genes and turn them into an actual heat map. Remember we did that? Well, actually, before we do that, okay, let's just get these genes out. Let's just create a file of the genes that we have in our results. So we'll take DSeq2 results. And we'll place them in a file. Let's call it DSeq2 results pdx.txt. Okay, let's add row names. And the row names are going to be from here, call names. Well, I don't know if we need call names. Let's just do separator by tab. And we'll add quote. That is the result from DSeq2. Okay, let's try this. Small mistake. Okay, what the volcano plot does is it has, well, let's see if we got our data set. So DSeq2 results PDX. So here's our result, right? So we generated that whole table of all of our differential genes. What is the volcano plot? So we have the two parameters. Remember the log fold change and the p-value. The p-value is the significance, and the statistical significance or confidence, and the log to fold change is the actual biological difference in terms of their gene expression. Now in log values, typically people say that anything above 2 or even 1 point something is going to be statistically significant. In terms of the p-value, it's going to be 0.5, right? So what we are trying to do here is we're trying to find the genes that have a significantly high fold change and a significantly low p-value. And remember that that could go both ways. Log fold change could be positive or negative. Log fold change means that it's the difference between the means of group 1 versus group 2. So it could be either 1 is low, 2 is high, or 2 is low, 1 is high. All right, so your assignment in terms of doing this on your own, and again, you can look through the exercises on here, differential gene expression. The objective is to find, or not find, but to actually extract the gene expression values, okay? So the gene expression values right here, this is not gene expression values, right? This is their statistical significance. So what you want to do is you want to find the statistically significant genes and find them in the original table in your PDX gene expression, find them in here, and then create a heat map, okay? So the objective is to create a heat map for these differentially expressed genes. Okay, so quickly about an additional element to remember here. In our case, when we have this kind of a comparison, we need to remember that the way that the data was created was actually created in a more complex way than just simply two conditions. And there are multiple examples of genes. If you actually look through the table, you'll see, if you have this additional information about which mouse model was used, these mouse models play a role, because the way that these mouse models were created to tolerate human tumors and grafted in them is by lowering their immune system so that the immune system does not reject these engrafted tumors. So in the log scale, fold change anywhere about 1.2 is probably going to be biologically significant. But depending on how many you have, you might want to make it higher, like 2. So when you have the real world situation, you have to be mindful of how additional factors about your data set play a role in your data. And to extract that information, you end up creating multiple hypotheses testing. And so the standard approach might be, well, let's just do everything, right? Let's do all of the comparisons between all of our pairwise comparisons. And you can do that with DESeq, but the problem is that it's going to be first useful to explore the actual conditions and the variation that you have. And so when we talk about our next session, we're going to start looking at a more in-depth exploratory analysis of our data, not looking at just the summary statistics or individual examples, but actually trying to summarize all of our data without losing any information, but really evaluating it for variance. And so here, an additional example, if you over the weekend have some time to look through Transcriptomics 2, you'll see that there's an additional analysis that is explained there called factor regression analysis. So the same way we looked at regression to study differences between or correlation with a particular between two particular samples, you can use correlation also to explore correlation with factors in your experiment design. And so please take a look at factor regression analysis. This is going to be in Transcriptomics 2. So Transcriptomics 1, you were able to prepare the table of expression. In Transcriptomics 2, you'll see here preparation of the data for downstream analysis, hypothesis testing, including differential gene expression, and factor regression analysis. And so that will talk a little bit about those. And remember, your assignment is to actually prepare a heat map of the differentially expressed genes. Okay, any questions about what we did today? On Monday, we will talk about outputs from the different pipelines that we started. You'll see some additional information that the pipelines give us. And what does that give us in terms of biological interpretation? How do we actually interpret the full results from a biological standpoint, not just from a statistically significant genes, but what do these genes actually do? And we'll start looking at principal component analysis. How do we apply principal component analysis to a few different examples? Now, actually, the second assignment that you have is to also try differential gene expression on another data set. Okay, so I'm just going to put this data set in here also. Okay, so let me just type this in and I'll put this on the forum as well. Okay, so assignment for Monday. 1. Explore or finish T1 and T2. Start T2, how about that? 2. Explore outputs from DE-seq 2 and try to interpret the findings. 3. Prepare a heat map for DE-seq 2 in R and apply DE-seq 2 to a different data set. In this data set, we will actually look at liver cancer and breast cancer and compare them. So this is going to be called this. Okay, any questions? All right, well, if there's no more questions, I'll be looking forward to seeing you on Monday, and have a great weekend, everyone.